Good to roll? Alrighty. Okay, good afternoon everyone. My name is Martin Chilvers, field crops pathologist here at Michigan State. Welcome to the Plant Path Farm. So we have a number of trials. I just figure I may as well explain what's going on here as well. So behind you we've got corn on corn ground that's been that way for about 10 years. We do a lot of seed treatment work. Over here we have been running a lot of tar spot work down in Decatur in southwest Michigan, but we brought a lot more of that back here, or duplicated some of those efforts. We have some residue that we're going to put out to help that get going. Over behind me here we have a soybean foliar block, so different foliar treatments that we're going to look at. And then back here, soybean seed treatment trials. And so we've challenged them with Pythium or Rhizoctonia. And in fact, a lot of that bare ground there is because of Rhizoctonia. So we've put that pathogen in the ground when we plant, stressing those seed treatments to see which ones work or which ones don't work. Just sort of related to wheat and cropping soybean after wheat. And I've been talking to Chris DeFonso about this a little bit. So up in here, this block was wheat last year. We did burn it down in the fall. We had some late tillage this spring and another burn down. So there was some weed residue, some wheat residue that was left over. But we have an awful lot of seed corn maggot damage out in there, which is really interesting to me. This trial that's flagged out with those 4 orange flags is one of those trials. So we've got an untreated and different biological treatments, and then Apron Max is our treatment. Anyway, I just think that's really interesting and a good use case for neonic seed treatments. You may have heard they've been in the news yet again for groundwater, well, not groundwater, but surface water contamination. So, I mean, maybe we need to pull back on use, but I would say that that could be a good use case where we really need these products. All right, so let's chat about wheat. If you guys have any questions on diseases or anything, please sing out as we go. I'd be happy to answer those. Most of our foliar fungicide trials back at the agronomy farm, we have a traveling linear system there to push water, to push head scab pressure and foliar disease. We've got a small block here. We've started some wheat seed treatment studies, and Bill Whitcomb has been building a seed or a planter for seed treatment trials. So we'll have that this coming fall. And there, in those trials, what we do is we take these different seed treatments and again examine them under different pressures. We'll put Pythium or Fusarium, whatever it is, down in the furrow when we plant, look at survival in that fall and through into spring. Anyway, it looks a little bit ratty, but that's what we have just happening over here, so some seed treatment work. Alrighty, so let's talk about a few other disease sort of issues. So head scab, I'm not aware of too much pressure around the state. We just sort of, I think, missed the window in terms of it was too cool. We didn't have that warm, wet conditions that are really favorable for head scab. In your packet or those handouts, you guys do have a fungicide efficacy chart. So every year we go through and update that. So based on new treatments that are coming through, new data that's collected here in Michigan and other states, we go ahead and update that efficacy information. So we'll rank products, be they good, excellent, or fair or poor, right, depending on performance. And similarly with fungicide efficacy testing for stripe rust and other foliar diseases. So that gets updated and we look at that, review it every year. Let's maybe pass around some cultures. Do you want to hand these out? Just distribute them out. Alrighty. We'll get that going. Well, just chat a little bit about stripe rust. Anyone have any stripe rust this year? A little bit, right? So we didn't have a great deal this year. So overall, we didn't see that buildup of inoculum down south and it moving forward and up north. And that's those years that we really run into a lot of trouble. So 2024, right, was pretty bad. And we had some pretty epic stripe rust out in this field. But it didn't develop here, it actually developed in Kurt Steinke's trials. He was earlier planted than we were, late planted, we just didn't seem to get it, and Christy Sprague had it early and late planted trials, and she saw differences there by planting date, which was really interesting. Something to do with the canopy and it catching spores and the right conditions for infection. Anyway, I did take a lot of spores from Kurt's study, sprayed them out here, and we had epic stripe rust data and pressure, so that was really helpful. So we just didn't get the build up. So we look for build up down south and then moving north every year. So that's really critical. 45 to 65 degrees for infection is somewhat important, and then for sporulation about 50 to 75 Fahrenheit. So there's all these sort of parameters that need to come together for those epidemics to really happen. I know some of the wheat farmers were over in the UK a couple of weeks ago, and it sounds like they had a failure of a resistance gene, so YR15. Did Eric talk about resistance genes? No, okay. So anyway, these resistance genes and the breeding work and monitoring for these is really important, right? To make sure we've got the right resistance genes in the varieties that you guys are growing to try and do a good job at managing. The only other thing that was really interesting with stripe rust this year is that my colleague in Ontario found it about level with a thumb in Ontario. So he was finding it early May, which if we found that here in Michigan, I'd be getting a little bit more concerned, right? Because that'd take us back to 24 and enough time for it to build up and really do some damage. So we don't know why it was there in Ontario. Again, I've been talking about it builds up down south, right in the country, and moves north. So maybe it overwintered, maybe we had the right amount of snowfall when temperatures were plummeting and it just allowed it to sort of survive. Anyway, so interesting and something that's, we're keeping an eye on. Again, with stripe rust, timely fungicide applications is absolutely key in those epidemic years. An early application, at Feeks 4, 5, 6, that's fine, but it might not be enough in those heavy years. We saw that in 2016 and 2024. You had to be on at flag leaf, and you couldn't wait until that head scab timing, right? Really important just to think about as we head into next year. All right, I'm going to hand this over to Keeley now. I guess before I do that, you guys want to put those plates in your luggage and head over to China? All right. So that's kind of what happened, right? So you guys might have heard all about that, no doubt. So I think the important thing to keep in mind is that this Fusarium is probably native to North America. There's been some studies done finding it on native grasses. It's globally distributed. I don't think these researchers at University of Michigan had any ill intent, right? They were just being maybe a bit lazy, bringing cultures over to work on. That lab looks at host pathogen interactions, which is a really important thing to do so we can better understand the disease and better manage it, right? But anyway, you can turn those back in if you want to. It is a Michigan strain that you have. I wouldn't be handing out strains even from like Illinois, Indiana, even though we share a border, right? That's not how the regulations work. If I want to bring in a strain, even across state lines, I have to have a permit to do that. And APHIS, USDA comes in and inspects us, make sure we have the way to kill it once we're done with working with it. So Keeley, this last couple of summers, has put in a lot of effort to some virus surveys across the state. So I'll have her just give us a little bit of a recap on that and what she's been finding.
Okay, so I'm Keeley. So for this year and last year, we've been doing a virus survey in wheat. And so I've been going through majority of the state, or Lower Peninsula at the very least. And last year I sampled 135 fields. This year I'm still having to work through things. So at least in your handout, it's saying I only have done 81 fields. And so in the fields, I sample along like edges, because that's how the vectors move in for the viruses. And then I sample symptomatic tissue to try to find viruses. And then we do the in-house testing in the lab and do an ELISA, which is an enzyme-linked immunosorbent assay. You don't have to remember that at all. It's just an immunological test to determine if there's a virus or in what specific type of virus. So I tested for 7 different viruses. They're listed in your handout. I will say them, but they're a lot. So I tested for barley yellow dwarf virus, cereal yellow dwarf virus, Bro mosaic virus, wheat spindle streak mosaic virus, soil-borne wheat mosaic virus, wheat streak mosaic virus, and high plains wheat mosaic virus. So I said that I sampled symptomatic tissue, and what that means is that I sampled for leaves that were like the symptoms of these viruses. So for cereal yellow dwarf and barley yellow dwarf, that means yellow tips or red or purple tips, and this is often on the flag leaf. And then like a green-yellow mosaic would be for like brown mosaic virus. And then a streaking yellow-green mosaic would be for like wheat spindle streak, wheat streak mosaic virus, and high plains wheat mosaic virus. So before I move into what I have been finding, I want to say that for the 2025 stuff, because I'm still going through it, it is subject to change, but that's what it was at the time when the paper was wrote. So for wheat streak mosaic virus, that's been the most prevalent virus for both this year and last year. It was found in 69% of fields last year and 91% of fields this year. And then barley yellow dwarf virus is the second most prevalent virus and was found in 61% of fields and 16% of fields this year. Cereal yellow dwarf virus is the third most prevalent virus and follows the same trend. The rest of the viruses, brown mosaic virus, wheat spindle streak mosaic virus, and high plains wheat mosaic virus were found infrequently. And then, surprisingly, soil-borne wheat mosaic virus wasn't found in any of the fields sampled, which is kind of interesting and fun. But what this means is that definitely need to kill volunteer wheat in neighboring fields. This is important since the three most prevalent viruses are spread by insects. Barley yellow dwarf virus and cereal yellow dwarf virus are spread by aphids, while wheat streak mosaic virus is spread by wheat chromites. And so these insects require a green living host after wheat is harvested. So volunteer wheat is the best choice in that because it's right there and it's growing and it's wonderful. So if you don't spray your volunteer wheat down, they can live all summer or the rest of the summer there, and then when fall happens, they can move into the fall planted wheat and infect those fields, which is important because fall infections is what causes the most yield loss. So spraying down volunteer wheat like 2 weeks before fall plantings is the best recommendation for management at the moment. Additionally, viruses are only managed by preventative measures, so variety resistance if possible. Some of these viruses, there's limited knowledge on the resistance for our soft winter wheat varieties, so take that as you will. And I want to finish with like 2 reminders and 1 suggestion. So my reminders is that viruses can't be controlled or managed by any pesticide, and that once a plant has a virus, there's no curable measures to be taken. Additionally, my suggestion is that if you don't know what something is in the field, I would send it in to get tested. I've been in fields where I'm like, oh, there'll be no viruses here, and sure enough, there's viruses. And virus symptoms look a lot like fertility problems and other stresses on plants, so it's always important if you're not quite sure to get it tested, especially since viruses are so tricky in that. So that was my spiel of my virus survey. If there's any questions, just let me know.
Thanks, Keeley. Thank you, Keeley. So as Keeley said, we've really got to manage the volunteer wheat. That's the important part here. It sounds kind of scary. Keeley's finding these viruses, in 60-80% of fields. Just because it's there doesn't mean it's doing a lot of damage. There's certainly many situations where these can cause a lot of issue, and typically where we've seen that is where we have a volunteer wheat field next to that next year's planting. So that's what we really want to keep in mind, and that's partly what we're sort of asking that next question of how much is actually doing damage. And work with Eric too to try and screen some lines here to look for resistance to those. Just in terms of identification as well, we had a little bit of a stripe rust scare early, but it turned out to be wheat streak mosaic virus. Again, if you're not sure what it is, get a sample into Jan at the diagnostic clinic. Really important thing to do that can save you thousands of dollars potentially, and make sure you're putting those inputs where you really need them. I guess, are there any questions, any pathology questions for us or comments? Nothing? You guys are quiet. I can make this awkward if you guys want. Yeah, Tom. Yeah, cover crop with oats could potentially be. Yes, potentially. We're sort of wondering about some of those, especially the grass species as cover crops. Absolutely, that could be a source. Yeah. Again, where we've seen the most damage is volunteer field next to wheat, and that's something else that definitely deserves additional study for sure. Probably, yeah. I mean, I think one of the other things to really think about here is that most of the transmission is probably happening in the fall, right? You see things in the spring, but the infection happened in the fall, right? It's a shame Chris DeFonzo's not here to talk about the insects a bit more, but Chris would emphasize we do not have any good miticides. We're not going to be spraying for these aphids either. So we've, again, just sort of got to think about those cultural practices. But those infections are going to happen in the fall. You might not see anything until the spring, and then, the field gets lit up. So something certainly to keep in mind. All right. Okay, if there's nothing else, I'll let you guys get rolling. Thank you very much.